to our YouTube channel AgriAddict. This is our YouTube lecture series for IBPS AFO mains exam, and also it is useful for all the other agricultural exams. I am your mentor Spoorthi, who have completed my BSc Hons Agriculture from University of Agriculture Sciences Bangalore, Subcampus VC Form Mandya. Currently, I am pursuing my Masters at ICAR IAB Ranchi under the Department Genetics and Plant Breeding. I have secured All India rank 6th in my ICAR PG exam of 2023-2024 in the subject Plant Sciences. Today, I am here to cover one of the most basic and important topic under genetics, that is history of genetics. Before learning anything about genetics, we have to study some of the historical events that are very important under genetics. Before diving into the topics, that is history of genetics, let us learn some of the basic terminologies which is most commonly used in genetics, in which the first and foremost it is the term genetics. As we know that genetics is a science which deals with the principles that explain the similarities between parents and their offsprings, which we call it as inheritance, and the difference among the individuals of single species, that is variation. That is, if we consider a species, for example human beings, there are certain similarities when we compare parents and the progenies, and also when we compare the individuals of the same human species, there are lots of variations. That is, there are certain differences from one person to another person. Genetics is the science which deals with the principles that explain why the similarities are observed between parents and the progenies, that is inheritance, and why the differences are present between the individuals of same species, and also when we compare to our parents there is lot of differences. So it explains why there is variation also. Simply we can call genetics as the science of inheritance and variation. Along with that, the term genetics is a Greek word which means to generate. Okay, genetics is derived from a Greek word which means to generate. Okay, now we'll study what is inheritance. Inheritance is nothing but it is the transmission of genes which is governing various characteristics from parents to progeny. That is, the genes which are responsible for various traits in an organism that is transferred from parents to the progeny. Okay, in that, what is character or trait? Character or trait is nothing but it is a feature which can be either morphological, anatomical, physiological, biochemical or behavioral of an organism. It can be any of the feature. It can be related to morphology or anatomy or physiology. That is called as character, or it is also called as trait. Next, coming to the most important term, that is genes. Genes are nothing but functional units which we can also call it as hereditary units that governs the development of various characteristics of an individual. Okay, see, if we consider a trait in plants, that is height, which is governed by, we know that it is governed by the gene T. This T represents gene. This gene T is responsible for the trait height in plants. Next term is allele. What is allele? It is the alternate forms of gene. For example, the plants can be either tall or it can be dwarf. This will be controlled by two alternate forms of this gene T, that is capital T and small t. That is, capital T codes for tallness and small t codes for dwarfness. This is the term alleles refers to. That is, they are the alternate form of genes. That is, the capital T which codes for the heightness is the gene, and their alternate forms are nothing but capital T and small t which codes for tallness and dwarfness respectively. Next, coming to genotype. As I said, consider if a plant which is tall in nature, then either it can be having the genotype capital T or it can be capital T small t. Since capital T is dominant in nature, it codes for tall. Whereas if the plant is dwarf in nature, then it can have only one phenotype, that is small t, small t. That is, since T is recessive in nature, it can express its character only when it is present in homozygous condition. Next, coming to the term phenotype. Phenotype is nothing but it is the external appearance of an individual, that what we see from outside, that is the phenotype. Whereas genotype, it is the genetic constitution of the organism, that is capital T or capital T small t represents the genotype, whereas the tall nature of the plant represents the phenotype. Okay. Next, coming to the branches of genetics. Genetics is nothing but it is the study of transmission of characteristics from one generation to another generation and variations. Similarly, cytogenetics. Cytogenetics is one of the branch of genetics which deals with various aspects of the chromosomes and their effects on development of characteristics of an organism. That is, we in detail study how chromosomes are present, what is the structure of chromosome, and how chromosome carries genes that will ultimately result in the development of an organism. That is, it is a very specific branch which deals with the chromosome. Next, coming to the branch cytology. In cytogenetics, we mainly focus on chromosome, whereas in case of cytology, we study the structural and functional organization of different structures present within a cell. That is, in total, we study entire cell, that is various organelles which are present in the cell, what are their function, how they are organized. We concentrate complete cell. Cyto means nothing but it is the cell, and logy which means study. So this study of cell. Next, one of the most important branch of genetics, that is molecular genetics. Molecular genetics will deal with the molecular basis of inheritance and variation, where in case of genetics we study how the genes are responsible for inheritance or variation, but in case of molecular genetics we study at the molecular level, that is at the DNA sequence level, how the DNA sequence are arranged and what a DNA sequence are made up of, how it is responsible for the inheritance and origin. That will be studied under the molecular genetics. Next, the two important genetics, that is forward genetics as well as reverse genetics. Okay, we'll study what is forward genetics. First, forward genetics. Forward genetics always begins with phenotype. Then we analyze the gene which is responsible for this particular phenotype, and then we analyze the DNA sequence of this particular gene. For example, if tall is the phenotype, then we identify the gene that is responsible for this tallness, that is capital T, and then we'll identify the DNA sequence of this tall gene, that is T gene. That is called forward genetics. That is, it begins with the phenotype of an organism and proceed to identify the genes or DNA that is responsible for that particular phenotype. Whereas in case of reverse genetics, that is, it always begins with DNA sequence whose function we are not known about. Like the DNA sequence, it codes for particular biochemical product. We'll identify the biochemical product that this DNA codes for. Then we'll study the phenotype that is produced because of this biochemical product. That is, it begins with the DNA sequence whose function is not known. Then it proceeds to determine its role in the development of phenotype. Forward genetics, always it begins with the phenotype, then we'll analyze the gene which is responsible for that phenotype, and then we'll analyze the DNA sequence. Whereas in case of reverse genetics, we start with the DNA sequence which function is not known, and then we'll determine the phenotype which is coded by them. These are some of the important terminologies that we have to study before diving it into history of genetics. Now we'll study history of genetics. The entire history of genetics has been classified into 4 different components. One, it is pre-Mendelian period, Mendelian period, post-Mendelian period, and also the genomics era, which can also be called as molecular era. Okay. The main basis for this type of classification is the period. Pre-Mendelian period deals with all the historical events before 1860. Next, Mendelian from 1860 to 1900. and from post-Mendelian period it is 1900 to 1944, and molecular era or the genomics era it is 1944 to present condition. Based upon the time period, we are classifying the entire historical events or history of genetics into pre-Mendelian period, post-Mendelian period and genomics era. Yes. Now we will study with respect to various historical events. Now I have listed down here some of the important historical events that has to be remembered from the exam point of view. Okay, everything started from 700 BC where Babylonians and Arsians, they cross-pollinated date palm in order to get good fruiting. That is the most basic thing that they started. They took 2 date palms and they cross-pollinated them in order to ensure the good fruiting. But later, one of the most important thing, that is in 1717, Thomas Fairchild, he was the one who was responsible for developing first artificial hybrid by crossing Carnation and Sweet William. This is an interspecific cross. And he named that hybrid as a Fairchild Mule. It was called as Mule because Mule stands for sterile. Even though he was able to successfully cross the Carnation and Sweet William, the resulting hybrid was completely sterile. Hence, it was named as Fairchild's Mule. And next, Janssen was the one who assembled the first microscope, and with the help of this microscope, de Graaf was able to discover the follicles in case of human's ovary, which is responsible for the production of the female gamete, that is egg. Next, Antony van Leeuwenhoek, he identified the sperms in animals, but he wrongly named it as animalculus. Next, Swammerdam along with Bonnet, they gave preformation theory. All the theories which are mentioned here will be explained in the last session. Okay, next, Grew, he was the one who identified that stamens are the male organs of the flowers which are responsible for the production of the male gamete, that is pollen grains. Then Cambrius, he identified and he discovered sexual reproduction in case of plants. Next, Cold-Reuter, even though the first artificial hybrid was developed in 1717 by Thomas Fairchild, Cold-Reuter later he was able to develop various hybrids in Nicotiana, Dianthus, Matthiola and Hyoscamus. Next, Lamarck. It has to be remembered that Lamarck, he proposed the theory of acquired characteristics that is popularly called as Lamarckism. This is a well-known theory where it says that the characteristics which are acquired by an individual during his life period will be transferred to the next generation even though it is not controlled by genes. This was proposed by Lamarck and it was also disproved, and one of the most popular example he gave it is the giraffe's neck. Next, coming to Amici, where he discovered pollen tube and the process of fertilization. How pollens will germinate on the stigma, how pollen tube penetrates stigma and enter into the ovary and fuses with the egg cell, and also process of fertilization in detail. Next, coming to O. Hartwig, where he studied fertilization in sea urchin. Next, coming to Darwin and Valence. This has to be remembered, that is, he proposed a theory of evolution through natural selection. That is how a particular species is undergoing evolution with the help of the natural selection, and also proposed the theory of pangenesis, which is also disproved. And it has to be remembered that Darwin, he published a book called as Origin of Species in order to explain the process of evolution. Later, Wolff, he disproved the theory of acquired characteristics that was given by Lamarck, and he proposed a theory called as theory of epigenesis. Next, coming to Brown, he was the one who first identified nucleus in case of Triscanthia plant. Next, these two important scientists, that is Schleiden and Schwann, they gave cell theory. Schleiden, he was the founder of cell theory in case of plants, whereas Schwann, he was responsible for cell theory in case of animals, and for which he is also known as father of modern cytology. Next, Janssen and Janssen together, they were responsible for assembling the first compound microscope. With the help of this compound microscope, Robert Hooke was able to identify and see cell in dead cork, and he also said that cells are nothing but they are the small compartment like structure. Okay. Next, coming to Nageli and Virchow, they postulated the third important postulate of the cell theory, that is all cells originates from the pre-existing cell. Next, coming to after this, it starts with the Mendelian era, that is from 1860 to 1900. Gregor John Mendel was the one who was responsible for proposing the laws of heredity, that is law of segregation and independent assortment, which was responsible for the inheritance genetics. These two laws are considered to be the principal laws upon which we are studying the classical genetics or the inheritance genetics, for which he is also called as father of genetics. Next, later Mischer was the one who isolated nucleic acid, but he called it as nuclein from the pus cells. When he isolated, it was the combination of both nucleic acid and protein. So he termed it as nuclein. But later Altman was successfully able to isolate only the nucleic acid part, that is free from the protein, hence he named it as nucleic acid. Next, Fleming was the one who coined the term mitosis chromatin, and he described the longitudinal division of chromosome during the cell division. That is, if consider this as an chromosome, this is transverse division, whereas this is the longitudinal division. If transverse division takes place, it results in the isochromosome, which is not true. He said that chromosomes always undergo longitudinal division in case of cell division, so that each of the sister chromatid reaches the opposite pole. And he also coined the term mitosis, which is a thread-like structure. Next, coming to Strasburger, he was the one who described meiosis in case of plants. Fleming was responsible for mitosis in case of animal cells, whereas Strasburger, he described meiosis in case of plants. Next, Farmer and Moore, they coined the term meiosis. Next, this is one of the most important event, that is in 1900, Hugo de Veres, Carl Correns and Teschmark independently they rediscovered the Mendelian theory. This was the major important event which was carried out by three different scientists independently. They were not together, they independently carried out the experiment. And Hugo de Veres, he is from Holland, and Carl Correns, he is from Germany, and Teschmark, he is from Austria. Try to remember the places from where they have come, and to independently they rediscovered the Mendelian theory. Next, later Waldier was the one who coined the term chromosome. Chromosome, it is derived from two words, that is chroma, which means color, and soma, which means body. Nothing but it is a colored body, because whenever we apply dye, then this chromosomes, since they are highly coiled structures, they absorb maximum dye and they appear as a colored body, hence it is called as chromosome. Next, coming to Pearson, where he gave chi-square test. Chi-square test, whenever we carry out any of the experiment related to Mendelian principles, in order to check whether the particular genes are following the laws of heredity, that is laws of segregation or independent assortment, or whether the two genes are linked together, we usually use the chi-square test. Next, Hugo de Veres, he gave mutational theory, that is how the mutations are responsible for the evolutionary process, whereas Darwin said that through the natural selection evolution takes place, but Hugo de Veres said that mutation is the ultimate cause that is responsible for the evolution. Next, coming to Landsteiner, he studied ABO blood groups, that is he said that the ABO blood group is controlled by single gene which is having 3 alleles, which is an example for the multiple alleles. And he said that even though there are 3 alleles, 4 different phenotypes are possible and 6 different genotypes are possible. Next, coming to McClung described the X chromosome in case of hemipterans insects. Later, Sutton and Boveri, they proposed chromosomal theory of inheritance. They were the first one who gave the concept that about the chromosomal theory of inheritance, that is genes are located on chromosomes. That is Sutton, he worked on grasshopper, and Boveri, he worked on sea urchin. but they were not able to give the experimental evidence. So they only said that yes, genes are located on the chromosome and behavior of this chromosome during the cell division is ultimately responsible for the Mendelian laws. But later it was proved by T.H. Morgan, that we'll see. Next, coming to Bateson. Bateson, he was the one who coined the term homozygote, heterozygote, allele epistatic gene F1, F2, and the term important, that is genetics. Okay, now we'll study what is homozygote. As I said that in case of tall plant, either it can have capital T genotype or it can have capital T small t. When two alleles are like each other, then it is called as homozygote. Whereas when two alleles are unlike, that is one is dominant and one is recessive, then it is heterozygote. Okay. In case of recessive characteristics, it only expresses and produces the phenotype when it is in the homozygote condition, whereas in the dominant allele, whenever it is present in homozygote as well as heterozygote, it produces the phenotype. Next, coming to Bateson and Punnett. Bateson and Punnett, they observed linkage in case of speed P, that is whenever we conduct a test cross. What is test cross? Test cross is nothing but cross between F1 into homozygous recessive parent. Whenever we conduct a test cross with respect to two genes, we usually get the ratio of 1:1:1:1. But when Bateson and Punnett, they conducted experiment using speed P with respect to the flower color and pollen length, they observed the test cross ratio of 7:1:1:7. That is, parental types were more. Hence, they said that the two genes responsible for the flower color and the pollen length were undergoing linkage, but same, they were also not able to give the experimental evidence for that. Next, Johansen was the one who gave the term gene, genotype, phenotype. As I explained, what is genotype and phenotype? Genotype is nothing but it is the genetic makeup of an organism, whereas phenotype is the external appearance. And also he proposed pure line theory in the Prince's variety of Rajma. This you will study with respect to the plant breeding classes. Like, remember that it was in the Prince's variety of the Rajma. Next, coming to Yule. Yule was the one who gave the basic idea of multiple factor hypothesis, where he said that there are certain quantitative characteristics which are controlled by multiple genes, and these multiple genes will have the cumulative effect. He gave the basic idea, but it was experimentally proven by Nilsson and Ale, where they used the seed color in wheats and oats, and they said that it is controlled by many genes. Next, later it was T.H. Morgan who gave sex linkage and proposed chromosomal theory of heredity in case of Drosophila. As I said that even though Sutton and Boveri, they said that chromosomal theory genes are located on the chromosome, but it was experimentally proven by T.H. Morgan using Drosophila, especially with respect to its eye color, and explained chromosomal theory of inheritance or hereditary. Later, one of his students in his lab, he named Sturtevant, he prepared the first ever linkage map of Drosophila, which has to be remembered. Next, Muller, that is H.J. Muller, he identified the mutagenic effects of X-rays in case of Drosophila. Similarly, Stadler, he identified the mutagenic effect of gamma rays and X-rays in barley and maize. In plants, it was by Stadler, and in case of Drosophila, it was Muller. Next, coming to Garrett. Garrett was the one who published Inborn Errors of Metabolism. He said that if any person who is suffering from genetic disorder, he will be lacking some of the enzymes which are required for particular biosynthetic pathway. That is, consider if a gene A is responsible for production of certain enzyme that is involving some biosynthetic pathway. That ultimately results in the phenotype. what Garrett said, if any mutation takes place, or if any person who is suffering from the hereditary disease, the particular gene will be hampered, so the enzyme that is required for the biosynthetic pathway will be absent, or it will be non-functional, so the particular phenotype will not be expressed, hence he will be suffering from the genetic disorder. And next, later Biddle and Tatum, they said that one gene, one enzyme hypothesis, for which they were also awarded with Nobel Prize in 1958. They said that one particular gene will codes for one enzyme, but later it was disproved because many enzymes are composed of many polypeptide genes. So later it was said that one gene will codes for one polypeptide. Next, coming to Avery, McCall and McCarthy, they were the one who said that DNA is the genetic material in Diplococcus pneumonia. Even though before this experiment, Griffith conducted the Griffith transformation experiment. Later, it was by Avery, McCall and McCarthy, they proved that DNA is the genetic material in the in vitro condition using Diplococcus pneumonia. Next, coming to Watson and Crick, one of the most important event, that is in 1953, they came up with the double helix model of the DNA, and the DNA type which they used is the B DNA. There are different types of DNA, examples like A DNA, B DNA, C DNA, Z DNA, which vary with respect to some of the parameters. But Watson and Crick, they used B DNA in order to explain the double helix model. Next, coming to McClinton. Barbara McClinton was the one who proposed the concept called ACDAC system in maize, which is commonly called as jumping genes. Jumping genes are nothing but these are the genes which are capable of transferring from one part of the genome to another part of the genome, through which they cause certain variations, in which the McClinton was able to identify ACDAC system in case of maize. Next, Nierenberg and co-workers, they were awarded with Nobel Prize for decoding the first triplet codon, that is UUU, which codes for phenylalanine. As we know that during the translation process, that is during the protein synthesis, the codons which are present on the mRNA will code for particular amino acid. This was identified by Nierenberg and co-workers, that is UUU will code for phenylalanine. Later, all the 64 different types of codons was discovered one after the other. Next, coming to Jacob and Menard, where they were awarded with Nobel Prize for operon model of gene regulation. In order to explain the operon model, if we consider this as a operon model where it consists of various genes, that is A gene, B gene, C gene, where each particular gene will code for one particular mRNA that will be converted into polypeptide, that is nothing but enzymes. In the similar way, B gene will produce one enzyme and C will produce one enzyme. Altogether, these enzymes will be involved in same metabolic pathway. Here, Jacob and Menard, they proposed lacoperon, where it was having three structural genes which coded different enzymes which were involved in the lactose metabolism, for which they were also awarded with the Nobel Prize. And later, Benzer was the one who gave the fine structure of R2 locus. Locus is nothing but the place where there is a presence of gene on the chromosome. If it is a chromosome, the point or the place where actually the gene is present, that is called as locus. He explained the R2 locus of T4 bacteriophage and coined the term cistron, recon and muton. Cistron is nothing but it is the place where there is a gene, and recon, it is the place where actually recombination takes place, and muton is the place where mutation takes place. Next, coming to Okovo and Korenberg, they were also awarded with Nobel Prize for in vitro synthesis of RNA and DNA respectively. Okovo was able to synthesize RNA in the laboratory conditions, and Korenberg was able to synthesize DNA in the laboratory conditions. Later, Smith, Nathans and Harbour, they isolated the first endonuclease enzyme, which completely changed the recombinant DNA technology, which was like evolutionary thing. What is this endonuclease enzyme? Endonuclease enzyme is the thing where they'll cut first we'll study restriction nucleases. Restriction nucleases are those enzymes which will cut the DNA at a specific position, at a restriction site. The site is called as restriction site, at a specific site, so that the gene of our interest can be easily taken out and it can be used for the recombinant DNA technology. There are two types. One is endonucleases and exonucleases. Exonucleases are those which cut the DNA at the extremes and at the terminals, whereas endonucleases, they cut the DNA in the inside part, where this type of enzymes are mostly useful in case of recombinant DNA technology. Consider if the gene of interest A is located here. If the restriction endonucleases are used, it cut the DNA at this position so that our gene of interest can be taken out and it can be used for the recombinant DNA technology. Next, later Crick, he gave Wobble hypothesis, that is during the translation process, whenever the tRNA is taking, like bringing the amino acids to the place where the protein synthesis takes place, the two codons is significant, whereas third codon is insignificant in nature. This was explained through Wobble hypothesis by Crick. Later, Baltimore and Temin, they identified the first reverse transcriptase enzyme. As we all know that is in central dogma, that is the information flows from DNA to RNA and RNA to protein. This we know. But there are certain cases, like in viruses, especially in case of retroviruses, RNA can be converted into DNA, which we call it as complementary DNA. Next, coming to August Weismann, where he proposed germplasm theory, which is the most accepted theory and it is presently utilized. Now, I have mentioned so many theories in the past slides, that is like preformation theory or theory of epigenesis or theory of acquired characteristics, and the most acceptable one, that is germplasm theory. Let us now study individual theories and understand its importance. The first and foremost theory that was proposed by Aristotle, it is theory of spontaneous generation. This theory says that all the living organisms which are present will originate from the dead matter, but it was disproved by Francis Reddy. Next, coming to same Aristotle, he also proposed preformation theory. Preformation theory says that there is a miniatures which are called as omniculus, which will be completely like human beings. It resembles human beings, but it is a miniature form which will be present either in eggs in females and sperms in males, and during the development of progenies, there is nothing but that miniatures will grow and give rise to a complete developed human beings, which is completely a wrong thing, and it was completely disproved. And later, theory of epigenesis, which was proposed by Wolff. Wolff says that if particular cell is present, it undergo continuous differentiation and growth and development in order to give rise to organs and tissues. This is accepted theory. That is, organs and tissues arise from a process of differentiation of a homogeneous embryonic tissues. Next, coming to theory of acquired characteristics, which is called as Lamarckism, given by Lamarck, where it says that the characteristics acquired by an individual in their generation will be transferred to next generation, even though it is not controlled by the genes, which is completely wrong. Next, coming to theory of pangenesis, which was given by Charles Darwin, where he says that each and every organ will have a invisible identical copy of itself, which is called as gemmules or pangenes, which will be transported to the gonads, that is the reproductive organs, during the gamete formation, so that it will be transferred to the gamete, and then fusion takes place, and then development of an organism takes place. It was also disproved. Next, coming to organic evolution or theory of evolution, which was given by Charles Darwin, where he says that two or more different species will evolve from a single parent species by undergoing variations with the help of the natural selection. That is, whoever is fittest to survive, that will be picked up by the nature, that is natural selection, and it will undergo continuous process of evolution. Next, the most accepted theory, that is germplasm theory, given by August Weismann, where he says that according to his theory, the body of an individual will have two types of tissue. One, it is somatoplasm, that is nothing but somatic cells, whereas second, it is the germplasm, that is germplasm. Somatic cells consist of all the body tissues which is not producing any gametes. These are like somatic cells. So any changes that takes place in somatic cells will not be transmitted to next generation. But in case of germplasm, it consists of germ cells, so it will give rise to gametes. So any changes in the gametes will be transmitted to the next generation. Next, coming to blending inheritance, genetic material from two parents will blend together in the progeny so that the character is having the intermediate phenotype between the two parents, which is also not true, because Mendel said that the genes that is factors which are responsible for inheritance is discrete in nature. That is, the genes which are obtained from the mother parent and the father parent, they won't mix together, they'll stay as it is and they will express themselves, which was also disproved. Next, coming to Mendelian inheritance, where he says that the traits are inherited according to his principles, that is law of segregation and law of independent assortment, which will be studied in the Mendelianism chapter. Next, coming to cell theory or cell lineage theory, which was given by Schleiden, Schwann, and the third important postulate was given by Nageli and Virchow. The important postulates under cell theory is that every life is composed of cells. That is, each and every organism except like viruses and other acellular organisms, all are composed of cells, and these cells are the structural and functional unit of the plants and animals. That has to be remembered. Cell is the structural and functional unit of plants and animals, and all the cells originate from the pre-existing cell. That is, single mother cell will undergo division to produce the daughter cells. In case of mitosis, 2 daughter cells, whereas in case of meiosis it will be 4 daughter cells. That is, all the cells will originate from the pre-existing cells. Then lastly, it is the chromosomal theory of inheritance, which was proposed by Sutton and Boveri, that says that genes are located on the chromosome, where they were not able to give it experimentally, but later T.H. Morgan, he proved experimentally using Drosophila. These are some of the important theories of evolution which explains how the particular evolution has taken place and how the organisms are organized and how the organism has evolved. These are the things with respect to the history of genetics, that is important historical events and also some of the important theories of evolution. If you have any doubts with respect to this topic, feel free to comment in the comment section. So our team will be very happy to solve your doubts. And I will take my opportunity to also say that AgriAddict has a Vijeta badge where it is exclusively meant for IBPS AFO exam, under which there is 50 plus standard mock test sets, previous year papers which are prepared by top rankers of ICAR PG exams, and it will also have one-on-one doubt clearing sessions also. Link for all those things will be present in the comment section. So if you have any doubts with respect to any of the topics I have covered today, please free to put a comment on the comment section so that we'll answer to that. So after this, the most important and exciting topic, that is cytology, that is with respect to cell ultrastructure. The part of the cell structure will be divided into two things, because it has to be studied in detail, because you will get many questions from the cell ultrastructure with respect to genetics. So thank you for watching this video and wait for the next chapter. Thank you.